Hello students, welcome to my channel. Now today I will discuss some important Icse questions from cell division and cell cycle. If you haven't watched the previous lessons on this topic, then first you can go and watch those videos because otherwise you will find problem while answering these questions. I have explained this topic in detail in all those videos. So let's start. Here is the first question. Now, you can pause the video and try to answer because in this way you can evaluate how much you have understood this topic. The first question is, rewrite the following terms in a correct and logical sequence. What are the terms? Interphase, anaphase, prophase, telophase, metaphase. Okay. Now, in my previous video, I have told about mitosis in detail, and these are the various stages of mitosis. At first, what happens? At first, a cell prepares itself so as to undergo the cell division or mitosis, and that preparation stage is interphase. So first interphase will come, right? Next, what will happen? The cell will enter into mitosis. And what is the first stage of mitosis? First stage of mitosis is prophase, because pro means first. After prophase, what stage comes? Metaphase, right? Meta means after. Then comes anaphase, right? And at the end, telophase comes. That is, telo means end, right? So first will be interphase, because here the cell prepares itself so as to undergo mitosis or the cell division. Then after preparing, it will enter mitosis. First stage of mitosis is prophase, then comes metaphase, then comes anaphase, and finally at the end is telophase. Clear? So this is how we will arrange these terms in a correct and in logical sequence. We will apply logic and we will rewrite these terms, or we will arrange these terms in a sequence. Clear? Now the next question. Next question, there are two differences. Okay, first difference is between cytokinesis and karyokinesis. Now, what is cytokinesis and what is karyokinesis? In my previous video, I have told you about both these terms. What is cytokinesis? Cytokinesis is the division of cytoplasm of the cell. It is the division of cytoplasm of the cell, right? And what is karyokinesis? Karyokinesis means all the nuclear changes that occurs in the cell during the cell division, or in one word you can say it is the division of nucleus. Okay, so cytokinesis is the division of cytoplasm and karyokinesis is the division of nucleus. You can say that all the nuclear changes that occurs in a cell during the cell division. Okay. Now, next difference. Difference between mitosis and meiosis, but here a criteria has been given on the basis of the number of daughter cells that are formed. What happens in mitosis and what happens in meiosis? That means how many number of daughter cells are formed. In case of mitosis, 2 daughter cells are formed, right? Whereas in case of meiosis, 4 daughter cells are formed. In mitosis, from 2n, we get 2n and 2n, these 2 daughter cells, right? In case of meiosis, what happens? 2n, then n gives to n, again n. So, we get 4 daughter cells in case of meiosis. But in case of mitosis, we get 2 daughter cells. As here the criteria is already mentioned, we have to differentiate on the basis of the number of daughter cells formed. That's why you have to be very specific while answering this type of question, because here the criteria is already mentioned, right? Now, next question. Now you have to read this question carefully. There is a diagram which has been provided. First look at the diagram and then try to answer the questions. First question is, identify the stage of the cell division. Look very carefully. This stage is anaphase. Why it is anaphase? See, these chromatids, they are far apart from each other. They have already been moved towards the opposite poles. This is one pole and this is another pole. Right? So they become far apart from each other and they have moved towards the opposite poles. Right? This is the characteristics of anaphase. So the stage of the cell division is anaphase. Clear? Now next question. Name the parts labeled A, B, C, D. 4 labelings you have to do. A, B, C and D. What is A? These dotted lines. These are the spindle fibers, right? I have already told you in my previous video. And what is B? The outermost layer. Cell wall. Why cell wall? Because this is a plant cell. Okay, this is a plant cell. There is a question at the end whether this is a plant cell or animal cell. I will give the answer in this particular question. Okay. Now what is C? Look very carefully. C is chromosome. Of course it's chromosome, right? And what is D? D is the centromere to which it's attached. See? D is this portion. D is centromere to which the sister chromatids get attached. Right? So C is chromosome and D is centromere. A is spindle fiber, B is cell wall. It is the outermost layer. Clear? These are the 4 labelings. Next question is, what is the unique feature that is observed in this stage? What is the unique feature? You can clearly see the unique feature by which you can say that this is the anaphase. Here the centromeres split and the chromosomes, they move apart from each other and they move towards the opposite poles, right? This is the unique feature. Okay. Clear? Now next question. Where does this type of cell division usually occur? What is the type of cell division that is occurring? Of course, it is mitosis that is occurring, right? Now, where does mitosis occur? In somatic cells, like in normal body cells. Okay, like in skin or in bone marrow, mitosis occurs. Clear? So you have to write that this type of cell division, that is mitosis, occurs in normal cells or somatic cells, like for example in skin. Right? Next question. How many daughter cells are formed from this type of cell division? I have already told you in the previous question. 2 daughter cells are produced, right, in mitosis. And next question is very important. Is the dividing cell shown a plant or an animal cell? And you have to give the reason to support your answer. This is a plant cell. Why? Because centrioles are not present. Here centrioles are not present. This is a unique feature of the plant cell. Centrioles are absent. Here centrioles are not present. That's why this is a plant cell. That's why B is cell wall, not cell membrane. Clear? Now move to the next question. Again, there are 2 differences. First difference is between centrosome and centromere. What is centrosome? Centrosome means it is an organelle of the cell. Centrosome is present only in animal cell, right? So it is an organelle of the animal cell. Clear? And what is centromere? Centromere is the region where the 2 chromatids of the, that means there are 2 chromatids present in the chromosome, right? So those 2 chromatids are joined with each other with the help of this centromere. So it is the central region of the chromosome. It is the central region of the chromosome. At that place, the chromatids are joined to each other. Right? Clear? Next is difference between chiasmata and crossing over. What is chiasmata? Chiasmata is the point of attachment of the non-sister chromatids of the homologous chromosome, where crossing over takes place. In my previous video, I have already told you that crossing over is the phenomenon. Okay. Crossing over is the phenomenon, but chiasmata is the point at which this phenomenon occurs. This is the X structure. Suppose this is the maternal chromosome and this is the paternal chromosome. This X structure where it is meeting, this point is chiasmata. And the phenomenon of this crossing, or this phenomenon of exchange of the genetic material between these two homologous chromosomes, that is the crossing over. So the point at which they get attached and this phenomenon occurs, the point is known as chiasmata, and the phenomenon of this exchange of the genetic material between non-sister chromatids of homologous chromosomes, that is crossing over. Clear? Now move to the next question. Now see the figure very carefully. First you have to name the stage. Look, what is the stage? This is very, very easy. Stage is metaphase. Why? Why metaphase? See, all the chromosomes are in equatorial plane. They all are lined up in the equator, right? This is the main characteristic of metaphase, right? So that's why this stage is metaphase. Now you have to label the parts from 1 to 4. What is 1? 1 is chromatid. Okay. What is 2? It's centromere. It's the middle portion where it gets joined. That means chromatids get joined. What is 3? 3 is centriole. This portion is centriole. And what is 4? 4 is the dotted line. That means they are the spindle fibers. Clear? These are the 4 labeling. 1 means chromatids. 2 is centromere. 3 is centriole. And 4 is spindle fiber. Clear? And how many chromosomes are shown here? Very easy question. 3 chromosomes. 1, 2, 3. So 3 chromosomes are shown here. Clear? And if a question comes that draw the next stage, this is metaphase. Draw the next stage. That means we have to draw anaphase. In the same way like it has been given in the question. And you have to maintain the chromosome number. So what you will do? Anaphase 1, 2, 3. Again 1, 2, 3. You have to maintain the chromosome number and the orientation that has been given in the question. Okay? Otherwise, marks will be deducted. Clear? Okay. So I think I have made myself clear and all these important questions are clear to you all. So this is for this lesson. I will upload a new video on some new topic very soon. Till then, stay tuned to my channel. And if you think my videos are informative and helpful, then like and share my videos. And don't forget to subscribe to my channel. Thank you for watching the video.